everybody. I first want to thank the organizer for giving me the opportunity to present you this research that deals with metal resistance genes in marine biofilms that colonize especially copper-releasing coatings. The context of our work is how metals, and especially copper, shape biofilm communities, and especially in the context of the Bay of Toulon, which is a highly anthropized bay due to numerous human activities, including large boat traffic and harbor activities or aquaculture, but also the French Navy. A previous study showed that planktonic microbial communities were shaped by trace element, but not the biofilm communities that seem to be less influenced by seawater concentration. We also work on antifouling coatings, which aim to limit the colonization process or biofouling on ship hull. Biofilm communities colonizing antifouling coatings based on copper control release were described as specific compared to non-biocidal substrates like plastics, for example. Our aim in this study was to identify drivers of biofilm community over a large scale, including 3 sites in temperate and tropical areas. Then to characterize the specificity of biofilms on metal-releasing coatings towards water or plastics. And finally, to determine if communities were enriched in metal gene resistance in these 3 dissimilar coastal sites and depending on the substrate. The 3 sites chosen were the Toulon Bay, which is our reference one. The second temperate location was Lorient Harbor, which is located in the northwestern coast of France in Brittany in the Atlantic Ocean. And the third one was located in the tropical Indian Ocean in La Réunion Island at Le Port. In addition to the measurement of physico-chemical parameters, microbial communities from the water column were sampled in 2 planktonic fractions, particle attached or PA, that represented microorganisms living attached to particulate organic matter, and free living or FL microbes. For biofilms, we used panels in triplicates that were immersed for 30 days at each location. PVC panels were used as a reference surface, and 2 copper-based antifouling paints were studied. HY released copper oxide, and A3 released copper thiosanate and zinc pyriton. Panels were scrapped and DNA extracted before the amplification and sequencing of the 16S RNA gene using a classical metabarcoding approach. Finally, qPCR was performed to quantify both the 16S RNA gene and 5 metal resistance genes for which primers were found in the literature. 3 were described as copper-resistant gene, COP-A, CUS-A, and QO. 1 as cobalt, zinc, and cadmium-resistant gene, and the last one as lead resistance gene. First results on biofilm and planktonic diversity were shown in this plot with form that stand for lifestyle and color for location. In addition to these 3 sites, biofilm from Bagnous, another site in the French Mediterranean coast, has been added in black. 3 clusters could be observed, indicating that biofilm communities on PVC had the highest richness and diversity compared to both particle-attached and free-living prokaryotes. Considering beta diversity, biofilm communities clustered separately from PA and FL, and the side effects appear less significant. A distance-based redundancy analysis performed with seawater parameters concluded that PA and FL were significantly driven by environmental parameters, especially nutrients and metals. However, when adding biofilm in the RDA, the percentage of explanation decreased drastically, indicating that biofilm were clearly less affected by environment than PA or FL. A discriminant analysis allows us to identify biomarker taxa for each lifestyle, with Planktomycete, Verrucomicrombia, anaerobic delta proteobacteria, or Bacteroidetes that were specific to biofilm communities. However, biomarker taxa are not always the most involved in specific function in the community. As we will see later, general Flavobacteriaceae and Altermonadaceae, for example, will be key taxa in biofilm on SPC. Also, they were identified as biomarkers of PA in blue. Considering only biofilms, color here stands for coatings with PVC in green, HY in red, and HY3 in violet. The density was lower on antifouling coatings as expected, except at La Réunion. We can also observe a significant decrease of the richness and diversity on the copper-based coatings in violet and red here. Multidimensional analysis based on weighted unifrac distance show that biofilm were mostly selected by surface, with all the PVC biofilm in green in the upper part of the plot, and only to a lesser degree by site. A huge selection seemed to occur due to the copper release on SPC. We assume that copper in contact with microorganisms trying to colonize SPC was much more considerable than copper dissolved in seawater that was shown to have limited effects on the biofilm in previous study. Considering site differentiation for surface, it was only due to close phylogenetic taxa, as shown by comparison between weighted unifrac and Curtis distances. The soft metacoder was used to display log 2 fold change of relative abundance of each taxon in pairwise comparisons between surface, which allow to identify specific taxa clearly selected by copper release coating. Planktomycetes, Verrucomicrobia, delta proteobacteria, or some Bacteroidetes were enriched in biofilms on PVC, so in green. Whereas Sphingomonadaceae, like Altererythrobacter, some gamma proteobacteria like Dazania or Alteromonas, and some Flavobacteriaceae like Aquamarina or Cycloclasticus were dominant in low diversified communities and SPC. So in violet. Considering metal resistance gene, Cus-Hay was representative of all the 5, with significant higher relative abundances on both HY and A3 compared to PVC at Lorient and La Réunion. Surprisingly, Toulon exhibited a clear different pattern with similar high level of Cus-Hay on all the surfaces, including PVC. Similar patterns were observed for the other genes. In addition, not all the MRG were present on PVC at Lorient and La Réunion, contrary to Toulon. Then we try to identify correlation between the relative abundance of biomarker taxa and metal-resistant genes. Copper resistance gene correlated with major biomarker like Dasania and Alteromonas on both species, but also with Myinobacter and Fabibacter, specifically on HY. CZCA was interestingly more associated with taxon A3, which is the pain that include zinc pyriton, while PBRT was also associated to A3 in Lorient and Toulon. Also, the highest seawater lead concentration was determined at La Réunion, indicating probably a lower specificity of this gene. To conclude, biofilm's community on artificial surfaces were significantly different from FL and PA. Alpha diversity increased from FL to PA and to biofilm, illustrating the integrative aspect of biofilm with little proportion of taxa shared with the two first. Beta diversity cluster first the lifestyles and then the sites. FL and PA were more affected by water quality, especially trace metals and nutrients, than biofilms. And we assume that EPS would be involved in this specific behavior. Metal-resisting surface composition had great influence on cell density, bacterial diversity, and composition, as well as on relative abundance of metal resistance genes. Specific taxa are present on metal-resisting surfaces independently from the site, but depending on metallic biocide composition. Cell tolerance or resistance to metals may be based on several emerging, with possible co-selection and not specific mechanisms. Finally, local contamination history may influence the basal level of energy enrichment in biofilm community. Then I want to thank especially the two postdocs that performed this work, so Elisa Catao and Thomas Paulet, and our collaborators in La Réunion and Lorient, and also in Banyuls. And finally, our financial support, the ANR and DGA. Thank you for your attention.